So here's something worth thinking about as you settle in tonight. Right now, as you're lying there, getting comfortable, maybe pulling the blanket up a little, your body is fighting a war, not a metaphorical one. A real, physical, ongoing conflict with millions of engagements happening every single second. Bacteria are trying to slip through tiny cracks in your skin. Viruses that hitched a ride on the air you breathed in an hour ago are bumping up against the wet lining of your throat, probing for a way inside a cell. And somewhere in your gut, a whole ecosystem of microorganisms is being kept in careful check, tolerated, but watched, like guests at a party where the host never quite takes their eye off the silverware. You don't feel any of this. That's the remarkable part. Your immune system is so good at what it does, so relentlessly competent, that most of the time you have absolutely no idea it's working. You go weeks, months, sometimes years without getting seriously sick, and you just sort of take that for granted. But underneath that calm surface, there's an engineering system more complex, more adaptive, and more elegant than anything human beings have ever built. And tonight, we're going to walk through it slowly, piece by piece, and try to understand how it actually works. Now, I want to be honest with you right up front. The immune system is confusing. It's genuinely, legitimately confusing, and not because you're missing something or because you didn't pay attention in biology class. It's confusing because it's a system that evolved over hundreds of millions of years, with no blueprint, no designer, no master plan. It's layers on top of layers, old solutions patched together with newer ones, redundancies built on top of redundancies. It's the biological equivalent of a city that's been continuously inhabited for 1,000 years, where Roman foundations sit beneath medieval walls, sit beneath modern plumbing, and somehow, impossibly, the whole thing still works. So if parts of this feel tangled or strange, that's not you. That's the system itself. And we're going to untangle it together, gently, with no rush at all. Let's start at the very beginning, and I mean the real beginning, long before anyone had any idea what an immune system even was. For most of human history, disease was a mystery, a terrifying, capricious mystery. People got sick, and nobody knew why. The ancient Egyptians, the Greeks, the Romans, the physicians of medieval Europe, they all had theories, but the theories were, by modern standards, completely wrong. The dominant idea for nearly 2,000 years was the humoral theory, which came primarily from a Greek physician named Hippocrates around 400 BCE and was later elaborated by Galen, a Roman physician working in the 2nd century. The basic idea was that your body contained 4 fundamental fluids: blood, phlegm, yellow bile, and black bile, and that disease happened when these fluids fell out of balance. Too much phlegm made you sluggish and cold. Too much yellow bile made you irritable and feverish. The cure was to restore balance, usually through bloodletting, purging, or dietary changes. Now, this sounds absurd to us, but here's the thing. It wasn't stupid. It was a genuine attempt to build a systematic framework for understanding the body, using the best observational tools available at the time. And some of the treatments actually worked, at least occasionally, even if the reasoning behind them was wrong. That happens more often in the history of medicine than you might expect, people stumbling onto the right answer for completely the wrong reasons. But there was one observation that kept nagging at people, one phenomenon that didn't quite fit neatly into the humoral framework, and it's the observation that eventually cracked open the whole field of immunology. People noticed that if you survived a disease, you often didn't get it again. This was not a subtle observation. It was obvious, dramatic, and consistent enough that people across multiple cultures picked up on it independently. The classic example is smallpox, one of the most devastating diseases in human history. Smallpox killed roughly 3 out of every 10 people who caught it, and those who survived were often left with terrible scarring. But the survivors had something precious. They almost never got smallpox a second time. And people noticed. They absolutely noticed. In 10th century China, physicians developed a practice called variolation, where they would take dried scabs from a person with a mild case of smallpox, grind them into a powder, and have a healthy person inhale it through the nose. The idea was to deliberately give someone a mild version of the disease so they'd be protected from the severe version later. Think about what that means. Think about the courage and the desperation and the careful observation that went into that practice. These physicians didn't know about viruses. They didn't know about antibodies or T cells or any of the mechanisms we're going to talk about tonight. But they had noticed a pattern, that exposure could create protection, and they had figured out a way to exploit it. That's engineering, in the deepest sense of the word. Solving a practical problem with the tools and knowledge available, even when you don't fully understand why the solution works. Variolation spread slowly westward over the centuries. It showed up in the Ottoman Empire, and from there it was brought to England in the early 1700s by Lady Mary Wortley Montagu, the wife of the British ambassador to Constantinople. She'd seen the practice in Turkey, had her own children variolated, and championed it back in London. This was not a smooth process. She faced enormous resistance, from physicians who were skeptical, from religious leaders who thought it was interfering with God's will, and from a general public that was understandably nervous about deliberately infecting themselves with one of the most feared diseases on earth. But she persisted, and gradually, variolation gained acceptance in parts of Europe. And then came Edward Jenner. You've probably heard of Jenner. He's usually the first name that comes up in any history of immunology, and for good reason, though the story is a bit more nuanced than the version most people know. Jenner was a country doctor in Gloucestershire, England, in the late 1700s, and he'd heard a piece of folk wisdom that was common among dairy workers, milkmaids who caught cowpox, a relatively mild disease they picked up from infected cows, seemed to be protected against smallpox afterward. Cowpox gave you a few sores on your hands, maybe a mild fever, and then it was over. But after that, smallpox couldn't touch you. Jenner decided to test this. In 1796, he took material from a cowpox sore on the hand of a milkmaid named Sarah Nelmes and scratched it into the arm of an 8-year-old boy named James Phipps. The boy developed a mild cowpox infection and recovered quickly. Then, about 6 weeks later, Jenner deliberately exposed him to smallpox, and nothing happened. The boy didn't get sick. He was immune. Now, by modern ethical standards, this experiment is horrifying. You cannot deliberately expose a child to a lethal disease to test a hypothesis. But in the context of the 1790s, with smallpox killing tens of thousands of people every year, Jenner's gamble made a kind of desperate sense. And the result was one of the most important medical breakthroughs in human history. Jenner called his technique vaccination, from vacca, the Latin word for cow. And that word, that idea, that principle, that you could train the body to fight a disease before it ever encountered the real thing, would eventually lead to the eradication of smallpox entirely, the last natural case occurring in Somalia in 1977. The complete elimination of a disease that had killed hundreds of millions of people. That's not a small thing. But here's what's fascinating. Jenner had no idea why vaccination worked. None. He knew that it worked. He could demonstrate that it worked. He could teach other physicians to do it. But the mechanism, what was actually happening inside the body when you introduced cowpox material and the body somehow learned to fight smallpox, that was a complete black box. And it would stay a black box for nearly another century. So let's sit with that for a moment, because it tells you something important about how understanding develops. There's often a huge gap between knowing that something works and knowing why it works. Engineers and physicians and scientists exploit this gap all the time. Aspirin was used for decades before anyone understood its mechanism of action. Builders used arches for thousands of years before the mathematics of structural mechanics could explain why they held up. And vaccination was practiced for 100 years before anyone could explain what an immune response actually was. The why started to come into focus in the second half of the 1800s, and it came from two directions at once, which makes the story wonderfully messy and human. The first direction was the germ theory of disease. Before the 1860s, most physicians still didn't accept that diseases were caused by microscopic organisms. The idea had been floating around for centuries. A few sharp observers had proposed it. But it wasn't until Louis Pasteur in France and Robert Koch in Germany started doing rigorous experimental work that germ theory became undeniable. Pasteur showed that fermentation and spoilage were caused by microorganisms. Koch developed methods to isolate specific bacteria and proved that they caused specific diseases: anthrax, tuberculosis, cholera. Between them, they demolished the old miasma theory, which held that diseases were caused by bad air, and established that the enemy was alive, tiny, invisible, alive, and everywhere. And once you knew the enemy was alive, the question became, how does the body fight it? What is the body actually doing when it encounters one of these microorganisms and defeats it? This is where we meet one of the most fascinating and underappreciated figures in the history of science. His name was Elie Metchnikoff, and he was, to put it gently, an unusual person. Metchnikoff was a Russian zoologist, brilliant and temperamental, who had already attempted suicide twice by the time he made his most important discovery. He was working at a marine biology station in Messina, Sicily, in 1882, studying starfish larvae under a microscope. His family had gone out. His wife had taken the children to the circus of all things. And Metchnikoff was alone in his laboratory, poking at translucent starfish larvae with a rose thorn. What he saw changed everything. When he pushed the thorn into the larva, he noticed that certain cells within the larva's body began to gather around the thorn. They migrated toward the foreign object and clustered around it, surrounding it, engulfing it. Metchnikoff realized he was watching the larva's body actively defending itself. These cells were eating the intruder. He called them phagocytes, from the Greek words for eating and cell. And he proposed something radical, that this process, this cellular eating, was the fundamental mechanism of immunity. That the body defended itself, not through some vague chemical process in the blood, but through specific cells that actively hunted down and consumed invaders. This was a huge idea, and it was also a hugely controversial one, because at almost exactly the same time, researchers in Germany, particularly Emil von Behring and Paul Ehrlich, were discovering something that seemed to tell a completely different story. They were finding that the liquid part of blood, the serum, contained substances that could neutralize toxins and kill bacteria even without any cells present. You could take blood from an animal that had recovered from diphtheria, remove all the cells, and the remaining fluid could still protect another animal from the disease. These protective substances would eventually be called antibodies, and the researchers studying them believed that immunity was fundamentally a chemical process, not a cellular one. So you had two camps: the cellularists, led by Mechnikov, who said immunity was about cells eating invaders, and the humoralists, a new kind of humoralism, very different from the ancient Greek version, who said immunity was about soluble substances in the blood neutralizing threats. And they argued about it, passionately, for years, with the kind of intensity that only scientists who are both brilliant and absolutely certain they're right can muster. And here's the beautiful thing, the thing that makes this story so satisfying. They were both right. Both mechanisms are real. Both are essential. Your immune system uses cells that physically engulf and destroy invaders, and it also produces antibodies that circulate in your blood and neutralize threats chemically. These two systems work together. They communicate with each other. They depend on each other. The argument between the cellularists and the humoralists wasn't really an argument about who was right and who was wrong. It was an argument between two groups of people who had each discovered one half of an enormously complex system and mistaken their half for the whole. Mechnikov and Ehrlich actually shared the Nobel Prize in Physiology or Medicine in 1908, which is one of those rare Nobel decisions that feels genuinely fair. Two rivals, two different approaches, both honored for contributions that turned out to be complementary rather than contradictory. But even with that recognition, the full picture of how the immune system works was still decades away from coming into focus, because the system Mechnikov and Ehrlich had begun to describe, this system of cells and antibodies, turned out to be only the more sophisticated half of an even larger story. There's a whole other layer of defense that's older, simpler, and in many ways more fundamental, a layer that your body shares with insects and plants and creatures that diverged from your evolutionary lineage hundreds of millions of years ago. And understanding that older layer, the one that's always on, always watching, always ready to respond within minutes rather than days, that's where we need to go next, because it's the foundation that everything else is built on. It's the part of your immune system that's working right now, this very second, as you lie there breathing quietly in the dark, and it doesn't even need to be told what to do. It just knows. It's been doing this job since before you were born, since before you took your first breath, since before your mother's body even finished building you. This is the innate immune system, and calling it innate is one of those scientific naming choices that actually makes intuitive sense for once. Innate means inborn. It means built in. It means you came into this world already equipped with it. No learning required. No prior exposure to any pathogen necessary. It's the immune system you get for free, just by being alive. And here's the thing. For most of the history of life on Earth, this was the only immune system there was. The adaptive immune system, the one with the antibodies and the specialized memory cells and all that sophisticated targeted machinery, that's a relatively recent evolutionary invention. It showed up maybe 400 or 500 million years ago, roughly around the time jawed vertebrates appeared. Which sounds ancient, and it is, but life itself has been around for close to 4 billion years. So for the vast majority of evolutionary history, organisms were defending themselves against infection using only innate immunity. Insects use it. Crabs use it. Starfish use it, which is exactly what Mechnikov was watching when he saw those cells swarm around the rose thorn. Plants have their own version of it. The innate immune system is one of the oldest, most conserved biological strategies on the planet, and the fact that it survived essentially unchanged across such wildly different branches of the tree of life tells you something important. It tells you it works. It works really, really well. So, let's talk about what it actually does. Let's say you get a small cut on your finger. Nothing dramatic, just a little nick while you're chopping vegetables. You might not even notice it at first, but in the moments after that cut happens, an extraordinary cascade of events begins. And it begins fast. We're talking seconds and minutes, not hours and days. The first thing that happens is purely mechanical. Your skin, which is itself the outermost barrier of your immune system, has been breached. And your skin is a genuinely impressive barrier. It's slightly acidic, which discourages bacterial growth. It's covered in antimicrobial peptides, these small proteins that can punch holes in bacterial cell membranes. It's home to its own ecosystem of friendly bacteria that crowd out potential invaders just by occupying space and consuming resources. Your skin is not just a wall. It's an active, hostile environment for anything that doesn't belong there. But now, there's a gap in that wall, and bacteria from the knife, from the cutting board, from the air, from the surface of your own skin are flooding into the warm, nutrient-rich tissue underneath. Almost immediately, cells that are already stationed in your tissue, cells that have been sitting there waiting for exactly this moment, detect that something is wrong. These are your sentinel cells, and the most important ones are called macrophages and dendritic cells. Macrophages. The name literally means big eater in Greek, which Mechnikov would have appreciated, are large, amoeba-like cells that patrol your tissues constantly. They're the descendants, conceptually speaking, of those very phagocytes Mechnikov watched clustering around the thorn in his starfish larva. When a macrophage encounters a bacterium, it engulfs it, just wraps around it, pulls it inside, and destroys it using enzymes and toxic chemicals contained in specialized compartments within the cell. It's brutal and efficient, and it happens without any instruction from the rest of the immune system. The macrophage doesn't need to be told that this bacterium is dangerous. It already knows. But how does it know? That's the question that puzzled immunologists for decades, and the answer didn't really start coming together until the 1990s, which is shockingly recent when you think about it. This is where a researcher named Charles Janeway enters the story, and honestly, Janeway deserves to be far more famous than he is. He was an immunologist at Yale, and in 1989, he gave a talk that would eventually reshape how we understand innate immunity. Janeway proposed something that seems obvious in retrospect, but was genuinely radical at the time. He suggested that the innate immune system has its own form of recognition, not the precise, laser-targeted recognition of antibodies, but a broader, more general kind. He argued that innate immune cells must carry receptors on their surfaces that can detect molecular patterns common to large groups of pathogens. Not the specific signature of one particular strain of bacteria, but the general features shared by whole categories of microorganisms. Think of it this way. The adaptive immune system, with its antibodies, works like facial recognition software. It can identify one specific individual out of millions. The innate immune system, by contrast, works more like a bouncer at a club who's been told to look for certain types of trouble. Anyone wearing a ski mask, anyone carrying a weapon, anyone who smells like smoke. The bouncer doesn't need to know the person's name. He just needs to recognize the warning signs. That's what Janeway was proposing. He called these molecular warning signs pathogen-associated molecular patterns, or PAMPs, and the receptors that detect them he called pattern recognition receptors, Now, Janeway's idea was elegant, but it was still largely theoretical when he proposed it. The actual receptors hadn't been found yet. That changed in 1996 when Jules Hofmann, a French biologist of Luxembourgish origin, made a remarkable discovery while studying fruit flies. Hofmann's lab had been investigating a gene in fruit flies called toll, which had originally been discovered in a completely different context, having to do with embryonic development. The name toll actually comes from the German word for amazing or crazy, because the researcher who first found the gene, Christian Nusslein-Volhard, reportedly exclaimed, "Das ist ja toll," when she saw the mutant fly embryos it produced. But Hofmann's group discovered that toll wasn't just involved in development. Flies with a mutated toll gene were extraordinarily susceptible to fungal infections. They couldn't fight off fungi that normal flies handled easily, which meant toll was playing a critical role in the fly's immune defense. This was a big deal. Here was a specific gene, a specific receptor, that the innate immune system was using to detect pathogens. And it was in a fruit fly, an organism with no adaptive immune system at all, no antibodies, no T cells, nothing but innate immunity. The toll receptor was exactly the kind of pattern recognition receptor Janeway had predicted. And then, the following year, in 1997, Janeway and his postdoctoral researcher, Ruslan Medjetov, identified a human version of the toll receptor. They called it toll-like receptor, or TLR, and they showed that when it was activated, it triggered inflammatory responses and helped activate the adaptive immune system. This was the bridge. This was the connection between the ancient innate system and the newer, adaptive one. The innate immune system wasn't just a crude first line of defense that held the fort until the real army showed up. It was the alarm system, the intelligence network, the decision maker that determined how and whether the adaptive immune system would respond. Janeway had been right. The innate system had its own sophisticated recognition machinery, and it was far more important than most immunologists had given it credit for. Hofmann would go on to share the Nobel Prize in 2011 for this work, along with Bruce Beutler, who identified the specific toll-like receptor in mammals that recognizes a component of bacterial cell walls called lipopolysaccharide. Janeway, tragically, had died of a brain tumor in 2003 before the Nobel was awarded. Many immunologists feel he should have been included. It's one of those bittersweet stories in science, the person who saw the shape of the answer before anyone else, who pointed everyone in the right direction, who didn't live long enough to receive the recognition that his insight deserved. But anyway, the point is this: your innate immune system is not dumb. It's not just a generic wall. It has receptors. We now know of at least 10 different toll-like receptors in humans, each one tuned to detect a different type of molecular pattern. TLR4 detects lipopolysaccharide from gram-negative bacteria. TLR3 detects double-stranded RNA, which is a hallmark of viral replication. TLR5 detects flagellin, the protein that makes up the whip-like tails bacteria use to swim. Each of these receptors is recognizing something that's essential to the pathogen's biology, something the pathogen can't easily change or disguise, because it needs that molecule to survive. It's a clever strategy. You don't try to recognize every individual enemy. You recognize the equipment they can't leave behind. So let's go back to that cut on your finger. The macrophages in your tissue detect the invading bacteria using their toll-like receptors and other pattern recognition receptors. They start engulfing and destroying bacteria. But they also do something equally important. They start sending out chemical signals. They release molecules called cytokines, which are essentially molecular alarm bells. These cytokines do several things at once. They make the blood vessels near the wound more permeable, so that fluid and immune cells from the bloodstream can leak into the tissue. They attract more immune cells to the site. They cause the local temperature to rise. That's inflammation, that redness and warmth and swelling you feel around a cut. Inflammation gets a bad reputation, and chronic inflammation is genuinely harmful, but acute inflammation at the site of an infection is exactly what you want. It's the immune system mobilizing resources. Among the first reinforcements to arrive are neutrophils, which are the most abundant white blood cells in your body. You have billions of them circulating in your blood at any given moment, and they live fast and die young. A typical neutrophil survives only about 5 days. They're like the rapid response team pouring out of the bloodstream and into the infected tissue in enormous numbers. Neutrophils are ferocious. They engulf bacteria like macrophages do, but they also release toxic granules and can even throw out their own DNA in web-like structures called neutrophil extracellular traps, NETs, that physically ensnare bacteria. When you see pus in an infected wound, what you're mostly looking at is dead neutrophils, millions of them, sacrificed in the fight. And all of this, every bit of it, happens without any involvement from the adaptive immune system. No antibodies needed, no prior exposure to this particular bacterium required. Your body has never encountered this exact strain before, and it doesn't matter. The innate system recognized the general category of threat and responded within minutes. For the majority of small infections you encounter in your life, this is enough. The innate immune system handles it, cleans it up, repairs the tissue, and you never even know anything happened. You might notice a little redness, a little swelling, maybe some tenderness, and then it's gone. The whole battle fought and won beneath your conscious awareness. But sometimes, and this is where the story gets really interesting, sometimes the innate system can't handle it alone. The infection is too large, or the pathogen is too virulent, or it has tricks to evade the innate defenses. And when that happens, the innate immune system does something remarkable. It calls for backup. It activates the adaptive immune system, that newer, more sophisticated layer of defense, with its antibodies and its memory cells, and its extraordinary ability to recognize specific individual threats with almost unbelievable precision. And the way it makes that call, the mechanism by which the ancient system hands off to the modern one, involves a particular type of cell that acts as a messenger between the two worlds, a cell that takes a piece of the invader, carries it to a lymph node, and presents it like evidence in a courtroom, saying, in effect, "This is what we're dealing with, and here's what it looks like. And now it's your turn to respond." And that cell, the dendritic cell, is one of the most elegant pieces of the entire immune system. Think of it this way. The dendritic cell has been sitting out there in your skin or in the lining of your gut, sampling the environment, and when it encounters something dangerous, when it picks up fragments of a bacterium or a virus, it doesn't just stay there and fight. It changes. It undergoes this transformation where it stops being a sentinel and becomes a courier. It pulls pieces of the pathogen apart, proteins really, broken down into small fragments called peptides, and it loads those fragments onto special molecules on its surface. These molecules are called MHC molecules, which stands for Major Histocompatibility Complex. And that's a mouthful, but the idea is beautifully simple. The dendritic cell is essentially holding up a piece of the enemy on a little molecular display shelf, showing it to anyone who comes close enough to look. And then it travels. It leaves the tissue where the infection is happening and migrates through the lymphatic system, those thin, clear vessels that run alongside your blood vessels, until it reaches a lymph node. You felt your lymph nodes before, probably. When you've been sick and noticed those tender, swollen lumps under your jaw or in your neck or your armpits, those are lymph nodes, and they're swollen because they're full of immune cells that are actively working. Lymph nodes are like meeting points, rendezvous locations, where the innate immune system comes to brief the adaptive immune system on what's happening out in the field. So the dendritic cell arrives at the lymph node carrying its molecular evidence, and now it needs to find the right responder. And here's where the adaptive immune system reveals its most astonishing feature, something that, when you really sit with it, is almost hard to believe. Inside your lymph nodes and circulating through your blood and lymph, you have billions of T cells and B cells. These are the soldiers of the adaptive immune system. And each one of them, each individual cell, carries a unique receptor on its surface, a receptor that recognizes one specific molecular shape, not a general category of threat, like the toll-like receptors we talked about earlier. One specific shape, one particular arrangement of atoms. Your body contains somewhere in the range of 1 billion different T cells, each with a slightly different receptor, each capable of recognizing a different fragment of a different pathogen. 1 billion different locks, waiting for their one specific key, and most of them will never find their match. Most T cells will circulate through your body for their entire lifespan and never encounter the exact molecular fragment they were built to recognize. They'll live and die without ever being activated, but the ones that do find their match, the ones whose receptor happens to fit the fragment being displayed by that dendritic cell, those cells wake up. They activate. And when they do, everything changes. This process is called clonal selection, and it was proposed in the late 1950s by an Australian immunologist named Frank Macfarlane Burnett. And Burnett's idea was radical for its time. Before him, the prevailing theory was something called the instructive theory, which suggested that antibodies were flexible molecules that could mold themselves around whatever antigen they encountered, like clay conforming to a shape. Burnett said no. He said the diversity already exists. He said your body pre-generates an enormous repertoire of different immune cells before you ever encounter a single pathogen, and then the pathogen selects which cells get activated. The antigen doesn't instruct the immune system how to respond. It selects from a pre-existing library. Think about what that means. Right now, sitting in your lymph nodes, you almost certainly have T cells and B cells that could recognize fragments of pathogens you've never been exposed to. Pathogens that might not even exist yet. Your immune system has, in a sense, pre-imagined threats it's never seen, generated receptors for molecular shapes that might only appear in a virus that evolves 10 years from now. It's not prophecy, it's probability. By generating enough random variation, the odds are good that something in your repertoire will be close enough to recognize almost anything. Burnett won the Nobel Prize in 1960 for this insight, shared with Peter Medawar, who had done groundbreaking work on immune tolerance, the equally important question of how the immune system learns not to attack your own body. And we'll come back to tolerance later, because it's one of the most important and most confusing aspects of the whole system. But for now, let's stay with what happens when that 1 in a billion T cell finds its match. The dendritic cell presents its fragment, the T cell's receptor fits, and now the T cell activates and begins to divide, rapidly, One cell becomes two, two become four, four become eight, and within days you can have tens of thousands of copies of that one specific T cell, all carrying the same receptor, all targeted against the same pathogen. This is clonal expansion, the clonal part of Burnett's clonal selection theory. You selected the right clone, and now you're expanding it into an army. But here's the thing. Not all T cells do the same job. There are different flavors, and the two most important ones to understand are helper T cells and killer T cells. Helper T cells, also called CD4 T cells, because of a molecule called CD4 on their surface, are the coordinators. They're like field commanders. When a helper T cell gets activated, it starts releasing cytokines, those signaling molecules we talked about during inflammation, and these cytokines do all sorts of things. They help activate B cells to start producing antibodies. They recruit more immune cells to the site of infection. They amp up the killing power of macrophages. They basically orchestrate the entire adaptive response. Without helper T cells, the adaptive immune system is essentially paralyzed. And this, by the way, is exactly why HIV is so devastating. HIV specifically infects and destroys CD4 helper T cells, gradually dismantling the immune system's command structure until the body can no longer coordinate a defense against even the most routine infections. Killer T cells, also called CD8 T cells or cytotoxic T cells, do exactly what their name suggests. They kill. But they kill in a very specific and targeted way. A killer T cell recognizes cells in your own body that have been infected by a virus. See, when a virus infects one of your cells, it hijacks the cell's machinery to make copies of itself, and in the process, fragments of viral proteins get displayed on the surface of the infected cell, again on those MHC molecules. The killer T cell comes along, recognizes that viral fragment, and delivers a lethal hit. It releases molecules called perforins and granzymes that punch holes in the infected cell's membrane and trigger it to self-destruct. The cell dies, and the viruses inside it die with it. It's targeted assassination at the cellular level. And then there are B cells. B cells are the antibody factories. When a B cell gets activated, usually with help from a helper T cell, it starts dividing and differentiating into what are called plasma cells, and plasma cells are extraordinary. A single plasma cell can pump out roughly 2,000 antibody molecules per second. These antibodies flood into the blood and lymph and tissues, and each one is shaped to grab onto the specific antigen that triggered the response. Antibodies work in several ways. They can coat a pathogen and mark it for destruction by other immune cells, a process called opsonization. They can clump pathogens together so they can't spread. They can block the specific molecular sites that a virus uses to enter your cells, effectively neutralizing it. They're versatile, precise, and produced in staggering quantities. Now, all of this, the clonal selection, the expansion, the differentiation into helpers and killers and plasma cells, takes time. This is why you get sick before you get better. The innate immune system responds in minutes, but the adaptive immune system needs days, typically somewhere between 4 and 7 days for a primary response, the first time you encounter a particular pathogen. During those days, the pathogen is replicating, the innate system is doing its best to hold the line, and you're lying in bed feeling miserable, running a fever, wondering why your body can't just fix this already. And the answer is that your body is fixing it. It's just building the specific tools it needs from scratch, and that takes time. But here's what's really beautiful about the adaptive immune system, and it's the thing that makes vaccination possible. After the infection is cleared, after the army of T cells and B cells has done its job and the pathogen is gone, most of those cells die off. You don't need tens of thousands of copies of that specific T cell anymore, but not all of them die. A small fraction, maybe 5 to 10%, survive and become memory cells. These memory cells are long-lived. Some of them persist for decades. Some of them, we think, might last your entire lifetime. And they're primed. If the same pathogen shows up again, these memory cells recognize it almost immediately. They don't need days to ramp up. They can mount a response in hours, not days. And the response is faster, stronger, and more effective than the first time around. This is why you generally only get chickenpox once. This is why vaccines work. A vaccine introduces your immune system to a harmless version of a pathogen, or a piece of one, so that your body can generate memory cells without you ever having to suffer through the actual disease. When the real pathogen shows up later, your immune system already has the blueprint. It already has memory cells on patrol, ready to expand at a moment's notice. The whole elegant cycle of recognition, activation, expansion, and memory. This is what Jenner stumbled onto in 1796 with his cowpox experiment, even though he had absolutely no idea about any of the cellular machinery that made it work. And speaking of that machinery, there's one piece of the puzzle we haven't talked about yet that might be the most mind-bending of all. The question of how your body generates all that diversity in the first place. How do you get 1 billion different T cell receptors and 1 billion different B cell receptors, each one unique, each one recognizing a different molecular shape? You don't have 1 billion different genes. Humans have roughly 20,000 protein-coding genes, which is not even close to enough to encode 1 billion different receptors. So how does the immune system do it? The answer was discovered by a Japanese scientist named Susumu Tonegawa, and it involved a mechanism so unexpected, so contrary to what biologists thought they knew about how genes work, that it fundamentally changed how we understand genetics itself. Tonegawa was working at the Basel Institute for Immunology in Switzerland in the 1970s, and the question he was wrestling with was exactly the one we just described. The prevailing dogma in molecular biology at that time was pretty straightforward. One gene encodes one protein. That's it. Clean, simple, elegant. And it worked beautifully for explaining hemoglobin, or insulin, or the enzymes in your liver. But when you tried to apply that logic to antibodies, the math just didn't work. If every unique antibody required its own dedicated gene, you'd need more genes just for your immune system than exist in your entire genome. Way more. And everyone knew that couldn't be right. But nobody could figure out the alternative. What Tonegawa discovered, through a series of brilliant experiments comparing the DNA in embryonic cells to the DNA in mature antibody-producing cells, was that the genes encoding antibody receptors aren't fixed. They're not sitting there in your genome as complete, finished blueprints. Instead, they exist as gene segments, little modular pieces that get physically rearranged, cut and shuffled and stitched back together in different combinations as each B cell matures. It's like having a wardrobe with 10 different shirt pieces, 10 different sleeve options, and 10 different collar styles. And each cell randomly picks one of each and sews them together into a unique garment. Except the numbers are bigger and the combinations are staggering. Here's how it actually works. The genes that encode the variable region of an antibody, the part that determines what shape it recognizes, are broken into segments called V, D, and J segments. In humans, there are roughly 65 V segments, 27 D segments, and 6 J segments for the heavy chain of an antibody alone. During B cell development in your bone marrow, each developing cell randomly selects one V, one D, and one J segment, and the cellular machinery literally cuts out the DNA in between and joins these segments together. This process is called VDJ recombination. And the key word there is random. Each cell makes its own independent random choices, so different cells end up with different combinations. Now, if you just multiply 65 times 27 times 6, you get about 10,000 combinations for the heavy chain. That's a lot, but it's not a billion. So where does the rest of the diversity come from? Several places, and this is where it gets really clever. First, there's a similar set of V and J segments for the light chain of the antibody, and those recombine independently. So you multiply the heavy chain combinations by the light chain combinations, and suddenly you're in the millions. But it goes further. The enzymes that do the cutting and joining are deliberately imprecise. They add or remove a few random nucleotides at the junctions where the segments are joined. This junctional diversity is essentially controlled sloppiness. Your cells are intentionally introducing errors at the splice points, and those errors create even more variation in the final protein shape. It's like if every time you sewed those shirt pieces together, the seam landed in a slightly different place, changing the fit of the garment in unpredictable ways. And then, for B cells specifically, there's yet another layer. After a B cell has been activated and is fighting an infection, it undergoes something called somatic hypermutation, where the genes encoding its antibody are deliberately mutated at an extremely high rate, about 1,000,000 times higher than the normal mutation rate in your other cells. The B cells whose mutations happen to improve their antibody's fit to the pathogen get selected and survive, while the ones whose mutations make the fit worse die off. It's evolution by natural selection, happening inside your body, in real time, over the course of days. And this is why the antibodies your body produces get better and better over the course of an infection. The first antibodies that show up are decent. The ones that arrive a week or two later are exquisitely precise, honed by round after round of mutation and selection. Tonegawa won the Nobel Prize in 1987 for discovering this gene rearrangement mechanism, and he won it alone, which is relatively unusual for the Nobel in physiology or medicine. The prize committee recognized that this wasn't just an immunology discovery. It was a fundamental revision of our understanding of how genes can work. Before Tonegawa, the genome was thought of as essentially static. You're born with a set of genes, and those genes stay the same in every cell of your body for your entire life. Tonegawa showed that in at least one critical system, that's not true. Your immune cells are actively rewriting their own DNA. They're editing their own genetic code to generate diversity. And that idea was genuinely revolutionary. So let's step back for a moment and appreciate the full picture of what's happening here, because it really is extraordinary. Your body generates billions of unique immune cells, each carrying a receptor that recognizes a different molecular shape. And it does this through a process of random genetic recombination that happens independently in each developing cell. Most of those cells will never encounter the pathogen their receptor matches. They'll patrol your body for a while and eventually die, never having been activated. But a tiny fraction of them, the ones whose receptors happen to match whatever pathogen you've been exposed to, those cells get activated, multiply, and mount a targeted response. It's an incredibly wasteful system in one sense. You're producing millions of cells you'll never need, but it's also incredibly robust because it means you're prepared for almost anything. Pathogens you've never encountered, pathogens that don't even exist yet. synthetic molecules that have never appeared in nature. Your immune system can recognize them all because it's not working from a list of known threats. It's working from a randomly generated library of shapes that's so vast it covers virtually every possibility. And here's where we need to talk about a problem that's been lurking beneath this whole beautiful system. A problem we flagged earlier when we mentioned Burnett's name. If your immune system randomly generates receptors that can recognize any molecular shape, then inevitably, some of those receptors are going to recognize your own body. Think about it. The recombination process is random. It doesn't know the difference between a bacterial protein and a human protein. So statistically, some of your T cells and B cells are going to end up with receptors that bind to molecules on your own healthy cells. And if those cells were allowed to activate and attack, you'd be destroying yourself. Your immune system would tear you apart from the inside. This is the problem of self-tolerance, and it's one of the most critical challenges the immune system has to solve. How do you maintain a library of billions of random receptors while making sure none of them attack you? The answer involves a process so ruthless it's almost hard to believe. It happens in the thymus for T cells, and it's the reason the thymus exists at all. Remember, we mentioned the thymus earlier, that small organ behind your breastbone that puzzled anatomists for centuries because they couldn't figure out what it did. Turns out the thymus is essentially a training camp and a killing field. It's where T cells go to be educated, and most of them don't survive the education. Here's what happens. Immature T cells, called thymocytes, are produced in the bone marrow and migrate to the thymus. In the thymus, they go through 2 rounds of selection. The first is called positive selection. The developing T cell is presented with MHC molecules, those molecular display cases we talked about, and the cell has to demonstrate that its receptor can actually interact with MHC. If it can't, it's useless, because T cells only work by reading peptides displayed on MHC molecules. So cells that fail positive selection, cells whose randomly generated receptors can't bind to MHC at all, are allowed to die. They just undergo programmed cell death, apoptosis, and they're cleared away. No drama, no inflammation. They just quietly disappear. But the second round is the really brutal one. It's called negative selection, and this is where tolerance is enforced. The T cells that passed positive selection, the ones that can interact with MHC, are now presented with self-antigens, proteins from your own body. And here's the remarkable part. The thymus has a special type of cell that expresses a gene called AIRE, autoimmune regulator, which was discovered in the late 1990s. What AIRE does is almost unbelievable. It causes thymic cells to express proteins from all over the body. Proteins that would normally only be found in the pancreas, or the eye, or the thyroid, or the skin. It's like the thymus creates a miniature molecular catalog of your entire body, a sampler platter of self, and developing T cells are tested against this catalog. If a T cell's receptor binds too strongly to any of these self-proteins, that cell is killed, deleted, eliminated before it ever leaves the thymus. The numbers here are staggering. Roughly 95 to 98% of all developing T cells die in the thymus. Let that sink in. Your body produces millions of T cells, and it kills the vast majority of them before they ever enter your bloodstream. Only the cells that pass both tests, capable of interacting with MHC but not reactive against self, are allowed to graduate and circulate through your body as mature, functional T cells. It's an incredibly stringent quality control process, and it's happening constantly, every day, right behind your breastbone. Peter Medawar, who shared that 1960 Nobel Prize with Burnett, was one of the key figures in establishing that tolerance is a learned process, not an inherent one. His experiments in the 1940s and 1950s showed that if you exposed an animal's immune system to foreign tissue very early in life, before the immune system was fully mature, the animal would accept that tissue as self and wouldn't reject it later. This was a stunning finding because it meant tolerance wasn't hardwired. It was something the immune system figured out during development, and it had enormous implications for organ transplantation, which is a whole story we should come back to. But here's the thing about thymic selection. It's not perfect. It can't be. If you deleted every T cell that had any reactivity to self, you'd probably eliminate too many useful cells, because there's some overlap between self molecules and pathogen-derived molecules. So the threshold is set to catch the most dangerous self-reactive cells, but some mildly self-reactive cells slip through. And for those cells, the body has backup mechanisms. Peripheral tolerance, it's called. Regulatory T cells, which are a special subset of CD4 T cells, actively patrol the body and suppress immune responses that shouldn't be happening. They're like the peacekeepers of the immune system, calming down other immune cells that are getting too aggressive or targeting the wrong things. When any of these tolerance mechanisms fail, when self-reactive cells escape deletion, or regulatory T cells don't do their job, or some environmental trigger pushes the system out of balance, the result is autoimmune disease. And autoimmune diseases are far more common than most people realize. Type 1 diabetes, where the immune system destroys the insulin-producing beta cells in the pancreas. Rheumatoid arthritis, where it attacks the lining of the joints. Multiple sclerosis, where it goes after the myelin sheath insulating nerve fibers. Lupus, where it can target almost any tissue in the body. In each case, the fundamental problem is the same. The immune system has lost the ability to distinguish self from non-self, and it's attacking the body it's supposed to protect. And this brings us to one of the most fascinating and still incompletely understood aspects of the immune system, the question of how it decides not just what to attack, but how intensely to respond, and when to stop. Because an immune response that's too weak leaves you vulnerable to infection. But an immune response that's too strong can be just as dangerous as the pathogen itself. And the history of medicine is full of cases where it wasn't the infection that killed people. It was their own immune system's overreaction. The most dramatic example of this might be something you've heard of, especially in recent years, called a cytokine storm. And the story of how we came to understand it takes us back to one of the deadliest events in human history, the 1918 influenza pandemic, and a mystery that puzzled scientists for decades. Because here's the thing about the 1918 flu. It didn't kill the people you'd expect. Most influenza outbreaks are hardest on the very young and the very old, the people with the weakest immune systems. But the 1918 pandemic had this terrifying W-shaped mortality curve, with a massive spike right in the middle, among healthy adults between the ages of 20 and 40. The strongest, most vigorous people were dying at the highest rates. And for a long time, nobody could fully explain why. The numbers themselves are staggering. Somewhere between 50 and 100 million people died worldwide, possibly more. It killed more people than World War I, which was still raging when the pandemic began. In some communities, especially remote ones with no prior exposure to influenza, the mortality rate was catastrophic. Entire villages in Alaska and the Pacific Islands were nearly wiped out. And the speed of it was unlike anything in modern memory. People would feel fine in the morning and be dead by evening. Their lungs would fill with fluid so quickly that they essentially drowned from the inside. Now, it took decades before researchers began to piece together what was really happening in those lungs, and the answer, when it finally came, was deeply unsettling. It wasn't just the virus destroying tissue. It was the immune system. Specifically, an overwhelming, uncontrolled flood of cytokines, those signaling molecules we talked about earlier, the ones that coordinate the immune response. In a normal infection, cytokines are essential. They call in reinforcements, they direct traffic, they amplify inflammation right where it's needed. But in the 1918 flu, something about the virus triggered an immune response so massive, so disproportionate, that the cytokines themselves became the weapon of destruction. Think about it this way. Imagine you have a small kitchen fire, and you call the fire department, which is exactly the right thing to do. But instead of sending one truck, they send every truck in the city. And every truck starts spraying water, not just at the fire, but at your entire house, your neighbor's house, the whole block. The fire gets put out, sure, but so does everything else. That's essentially what a cytokine storm does to your lungs. The inflammatory response becomes so intense that it destroys the very tissue it's trying to protect. The blood vessels in the lungs become leaky. Fluid pours into the air spaces. Immune cells flood in by the millions, releasing destructive enzymes and reactive oxygen species, and the delicate architecture of the lung, those incredibly thin membranes where oxygen passes into your blood, gets shredded. And this is why the 1918 flu killed young, healthy adults at such alarming rates. It's precisely because their immune systems were strong. A robust, well-functioning immune system mounted a more powerful cytokine response, which meant more inflammation, more tissue damage, more fluid in the lungs. The very thing that normally protects you, a vigorous immune response, became the thing that killed you. It's one of the most profound paradoxes in all of immunology. Now, understanding cytokine storms didn't happen overnight. The term itself didn't really enter common medical usage until the 1990s, and even then it was mainly discussed in the context of certain rare conditions. Graft versus host disease after bone marrow transplants, some severe viral infections, and a few cases involving experimental treatments. But the concept became dramatically, urgently relevant again in the early 2000s, and then again in ways that most of us remember very personally. In 2003, the SARS outbreak, caused by a coronavirus, a close relative of the virus that would later cause COVID-19, showed clear evidence of cytokine storm pathology in its most severe cases. And then, in 2005, something happened in a laboratory that drove the point home in the most vivid possible way. A team led by Jeffrey Taubenberger at the Armed Forces Institute of Pathology, along with Terence Tumpy at the CDC, did something extraordinary and, some would argue, terrifyingly risky. They reconstructed the 1918 influenza virus. They actually rebuilt it from scratch, using genetic sequences recovered from preserved tissue samples, including lung tissue from an Inuit woman who had been buried in permafrost in Alaska since 1918, her body preserved by the frozen ground for nearly 90 years. When they infected mice with this reconstructed virus, the results were dramatic. The mice mounted enormous immune responses, massive cytokine production, severe lung inflammation, and they died quickly. The virus itself was devastating, yes, but the immune overreaction was clearly a major part of what made it so lethal. It was confirmation, nearly a century later, of what clinicians in 1918 had observed but couldn't explain, that the body's own defenses were contributing to the destruction. And here's where this connects to something much more recent, something that probably affected your life directly. When COVID-19 emerged in late 2019 and swept across the world in 2020, one of the most dangerous complications in severe cases was, once again, a cytokine storm. The pattern was eerily similar. Patients would seem to be managing the initial infection, and then suddenly deteriorate as their immune system went into overdrive. The lungs would fill with fluid, organ systems would start failing, and the treatment, paradoxically, often involved suppressing the immune system rather than boosting it. Drugs like dexamethasone, a corticosteroid that dampens immune activity, turned out to be one of the most effective treatments for severe COVID. Think about how counterintuitive that is. You're fighting an infection, and the best thing doctors can do is partially shut down your immune response. But it makes perfect sense once you understand that the immune system can be just as dangerous as the pathogen. This whole idea that the immune system needs to be regulated, modulated, sometimes even restrained, is one of the great themes of modern immunology, and it connects to something we should talk about now, because it's one of the most consequential medical breakthroughs of the 20th century, and it starts with a gruesome problem that surgeons had been wrestling with for centuries: transplantation. The idea of replacing a damaged organ with a healthy one is ancient. There are medieval paintings of saints miraculously transplanting limbs, but as a practical medical reality, it was essentially impossible until the mid-20th century, and the reason wasn't surgical technique. Surgeons figured out how to physically connect blood vessels and sew organs into place surprisingly early. The French surgeon Alexis Carrel won the Nobel Prize in 1912 for developing techniques for vascular suturing, basically, how to sew blood vessels together. Technically, you could transplant a kidney by the early 1900s. The surgery itself was solvable. The problem was what happened afterward. The body rejected the transplant every time. The immune system recognized the new organ as foreign, as non-self, and attacked it with the same ferocity it would direct at a dangerous pathogen. The transplanted organ would become inflamed, its blood supply would be choked off, and within days or weeks it would die. And remember what we talked about earlier, Medawwar's work during World War II, studying why skin grafts between different individuals always failed? That was the grueling experimental proof that rejection is an immune phenomenon, that the body learns what is self and treats everything else as a threat. Medawar shared the 1960 Nobel Prize with Burnett for that work on acquired immunological tolerance, and it laid the intellectual foundation for everything that came next. But knowing why rejection happens and knowing how to prevent it are very different things. The grueling clinical reality was that you could perform a technically perfect transplant surgery and watch the organ be destroyed by the patient's own immune system. And for years, the only tools available to suppress that immune response were blunt and dangerous: whole body irradiation, which destroyed the immune system, but also destroyed the bone marrow and left patients vulnerable to every infection imaginable, or drugs like azathioprine and corticosteroids, which broadly suppressed immune function, but with severe side effects and inconsistent results. The first truly successful kidney transplant happened in 1954, performed by Joseph Murray at Peter Bent Brigham Hospital in Boston, and the reason it worked tells you everything about the problem. The donor and recipient were identical twins, Richard and Ronald Herrick. Because they were genetically identical, Richard's immune system recognized Ronald's kidney as self. There was no rejection. Ronald lived for 8 more years with his brother's kidney, and Murray eventually won the Nobel Prize in 1990. But the success of that surgery also underscored the limitation. Transplantation only worked when you could cheat the immune system by using a genetically identical donor. For everyone else, rejection remained the wall. The breakthrough that changed everything came from a place you might not expect: a soil sample from Norway. In the early 1970s, a researcher at Sandoz Pharmaceuticals named Jean-François Borel was screening compounds derived from fungi for potential medical uses. One of the compounds came from a fungus called Talipocladium inflatum, found in a soil sample collected from the Hardangervidda Plateau in Norway. The compound was cyclosporine, and when Borel tested it, he discovered something remarkable. It suppressed the immune system, but in a surprisingly targeted way. Instead of broadly wiping out all immune function, cyclosporin primarily inhibited T cells. Specifically, it blocked the production of interleukin-2, a key cytokine that T cells need to proliferate and mount an attack. It was still an immunosuppressant, still carried risks, but it was far more selective than anything that had come before. Cyclosporine entered clinical use in the early 1980s, and the effect on transplant medicine was transformative. Suddenly, kidney transplants from unrelated donors had dramatically better survival rates. Heart transplants, which had been attempted with limited success since Christian Barnard's famous first in Cape Town in 1967, became genuinely viable. Liver transplants, lung transplants, procedures that had been experimental and often fatal became routine. The one-year survival rate for kidney transplants jumped from around 50% to over 80%. It's hard to overstate how much of modern transplant medicine rests on this single compound, isolated from Norwegian dirt. But cyclosporine also illuminated a deeper truth about the immune system, one that connects back to everything we've been talking about. The immune system isn't a single on-off switch. It's a network of checks and balances, signals and countersignals, accelerators and brakes. And the art of medicine, whether you're treating autoimmune disease, managing a transplant, or fighting a cytokine storm, is increasingly about learning to adjust those dials with precision, not shutting the system down entirely, not leaving it unchecked, but finding that narrow window where it does exactly what you need, and nothing more. And this idea of precision, of targeting specific parts of the immune system rather than hammering the whole thing, is what drives one of the most exciting and still rapidly evolving areas of medicine today. Because in the last few decades, scientists have figured out how to do something that would have seemed like science fiction to Jenner, or Pasteur, or even Medawar. They've learned how to engineer the immune system itself, to reprogram it, to turn it into a weapon against diseases that have nothing to do with infection. And the story of how that happened starts with a grueling, decades-long effort to answer a question that had haunted oncologists since the very beginning of cancer medicine. A question that sounds almost naive when you first hear it, but turns out to be one of the most profound in all of biology. Why doesn't the immune system just destroy cancer the way it destroys a virus or a bacterium? Think about it. Your immune system can detect a single flu-infected cell among billions of healthy ones, mobilize an army of T cells and antibodies, hunt that infection down, and wipe it out in a week or two. So why does it let a tumor, which is, after all, your own cells growing out of control, just sit there and grow? Why doesn't the immune system notice? Well, here's the thing. It does notice. Sometimes. And that sometimes is what makes the whole story so fascinating and so frustrating. The idea that the immune system might play a role in fighting cancer is actually really old. It goes back to at least the 1890s, to a New York surgeon named William Coley. And Coley's story is one of those beautiful, strange detours in medical history, where someone stumbles onto something genuinely important, gets it partly right, gets dismissed by almost everyone, and then, a century later, turns out to have been onto something profound. Coley was a bone surgeon at what's now Memorial Sloan Kettering Cancer Center, and in 1891, he had a patient, a young woman named Bessie Daschle, who came in with a sarcoma in her arm. He operated, but the cancer came back, and she died. Coley was devastated. He was young, just starting his career, and this case haunted him. So he did something that a lot of great scientists do when they're stuck. He went digging through old medical records. He started combing through case files at the hospital, looking for anything that might give him a clue about what made some cancer patients survive and others die. And buried in the records, he found something remarkable. A patient named Fred Stein had come in years earlier with a sarcoma in his neck. The tumor was inoperable. But then Stein developed a severe skin infection, erysipelas, caused by Streptococcus bacteria. And when the infection finally cleared, his tumor had shrunk dramatically. In fact, it essentially disappeared. Stein walked out of the hospital cancer-free. Coley tracked Stein down, actually found him alive and well, living in New York years later. And that convinced him. He started deliberately injecting cancer patients with bacterial toxins, heat-killed bacteria, mixtures of Streptococcus and Serratia, what became known as Coley's toxins, and some of his patients got better. Not all of them, not even most of them, but some of them showed remarkable tumor regressions. Fevers would spike, the body would mount this massive inflammatory response, and tumors would shrink. The medical establishment largely dismissed him. This was the era when radiation therapy was coming into its own. The Curies had discovered radium. X-rays were being used to zap tumors, and that felt scientific, precise, modern. Injecting people with bacterial soup felt like quackery. After Coley died in 1936, his approach was mostly forgotten. But here's what's wild. Coley was right about the core insight. He just didn't have the tools to understand why it worked. What was actually happening was that his bacterial toxins were activating the immune system. The same innate immune pathways we talked about earlier, the toll-like receptors, the pattern recognition molecules, and that activated immune system was, in some cases, turning against the tumor. He was doing immunotherapy before anyone had a word for it. The formal theoretical framework didn't come until the 1950s and 60s. In 1957, a brilliant Australian immunologist named Macfarlane Burnet, the same Burnet who gave us clonal selection theory, proposed something he called immune surveillance. The idea was elegant. Your immune system is constantly patrolling your body, looking for cells that have become cancerous, and most of the time, it finds them and kills them before they ever become a detectable tumor. The cancers that do grow, in this view, are the ones that somehow slipped through the surveillance net. It was a beautiful hypothesis, and for a while, it seemed to be supported by the evidence. Transplant patients on immunosuppressive drugs, people taking cyclosporine and similar medications, did develop higher rates of certain cancers. That made sense if the immune system was normally keeping those cancers in check. But then the theory ran into trouble. In the 1970s and 80s, researchers bred mice that lacked functional immune systems. Nude mice, they were called, because they also lacked fur, and expected these mice to be riddled with spontaneous tumors. But they weren't. Their cancer rates weren't dramatically higher than normal mice. That seemed to undercut immune surveillance pretty severely, and the whole idea fell out of favor for a couple of decades. It took until the late 1990s and early 2000s for the concept to come roaring back, refined and updated. Robert Schreiber at Washington University, along with Lloyd Old and others, proposed a more nuanced model they called cancer immunoediting. And this is really the modern understanding. So, let's sit with it for a moment. The idea is that the relationship between your immune system and a developing cancer goes through 3 phases. They called them the 3 E's: elimination, equilibrium, and escape. In the elimination phase, the immune system does exactly what Burnett predicted. It detects abnormal cells and destroys them. This is happening in your body right now, probably. Cells occasionally pick up mutations that push them toward uncontrolled growth, and your T cells, particularly your CD8 killer T cells and your natural killer cells, recognize that something's wrong and take those cells out. You never know it happened. Most potential cancers die in this phase, but sometimes some cancer cells survive. Maybe they're not quite abnormal enough to trigger a strong immune response. Maybe they're in a location where immune surveillance is less active. Whatever the reason, they persist. But they don't grow freely either. The immune system keeps them in check, holding them in a kind of stalemate. This is the equilibrium phase, and it can last for years, maybe decades. You're walking around with a tiny cluster of abnormal cells that your immune system is suppressing, but not eliminating. Think about how strange that is. You might have a cancer right now that your immune system is sitting on, keeping it contained, and you'll never know about it unless something changes. And that's the 3rd E, escape. Over time, through the relentless pressure of natural selection, the cancer cells that are best at evading the immune system survive and proliferate. They evolve right there inside your body. They develop tricks. And this is where it gets really interesting, because the tricks cancer cells use to escape immune detection are incredibly sophisticated. Some of them stop displaying MHC molecules on their surface. Remember those molecular display cases we talked about? The ones that show fragments of internal proteins to passing T cells? If a cancer cell downregulates its MHC expression, it becomes essentially invisible to CD8 T cells. It's like a criminal removing the house numbers so the police can't find the address. Other cancer cells go further. They don't just hide, they actively suppress the immune response. They secrete chemical signals that tell nearby T cells to stand down. They recruit regulatory T cells, those same tregs that normally prevent autoimmune disease, and co-opt them into creating an immunosuppressive environment around the tumor. The tumor essentially builds a protective bubble, a microenvironment where immune cells enter but lose their ability to fight. And here's the kicker. One of the most important escape mechanisms involves something called immune checkpoints. This is the discovery that changed everything, and it starts with a researcher named James Allison. Allison grew up in Alice, Texas, a small town in the southern part of the state. His mother died of lymphoma when he was 11. That loss shaped him, though he said he didn't consciously decide to fight cancer because of it. He was drawn to basic science, to understanding how T cells work, not to clinical medicine. But the thread is there. In the 1990s, Allison was studying a molecule on the surface of T cells called CTLA4. Now, by this point, immunologists knew that T cells need 2 signals to become fully activated: the antigen recognition signal through the T cell receptor and a second co-stimulatory signal, kind of like a confirmation code. CTLA4 was known to be involved in this process, but its exact role was debated. Some researchers thought it was an activator. Allison became convinced it was the opposite, a brake. CTLA4, he believed, was an off switch. When a T cell was activated and doing its job, CTLA4 would eventually engage and tell the T cell to calm down, to stop attacking. It was part of that system of checks and balances we keep coming back to, a safeguard against the immune system overreacting. And then, Allison had the insight that made history. If CTLA4 is a brake on T cells, and if cancer is partly a failure of T cells to attack tumors aggressively enough, what happens if you release the brake? What if you block CTLA4 and let the T cells go full throttle? In 1996, his lab tested this in mice. They created an antibody that blocked CTLA4, injected it into mice with tumors, and the results were stunning. The tumors shrank. In many cases, they disappeared entirely. The mice were cured, not by attacking the cancer directly, but by removing a constraint on the immune system and letting it do what it had been trying to do all along. Now, getting from mice to humans took years. Allison spent the better part of a decade pushing pharmaceutical companies to develop a human version of his anti-CTLA4 antibody. It wasn't easy. The drug industry was skeptical of immunotherapy. Decades of failed cancer vaccine trials had made people cynical. But eventually, a drug called ipilimumab was developed and tested in patients with metastatic melanoma, one of the deadliest forms of skin cancer. In 2010, the results of a major clinical trial were presented, and for the first time in history, a treatment was shown to extend survival in advanced melanoma. Not by months. Some patients were still alive years later. Some appeared to be cured. In a disease where the median survival had been measured in months, this was extraordinary. Meanwhile, on the other side of the Pacific, well, actually, just across the ocean of academic disciplines, a Japanese immunologist named Tasuku Honjo had discovered another checkpoint molecule, this one called PD-1. Honjo found PD-1 in 1992, and like Allison's CTLA4, it turned out to be a brake on T cells, though it works through a different mechanism and at a different stage of the immune response. Cancer cells, it turned out, had evolved to exploit PD-1 brilliantly. Many tumors express a molecule called PDL1 on their surface, the ligand for PD-1, which essentially sends a "don't kill me" signal to any T cell that comes close. The cancer cell is actively pressing the brake pedal on the very immune cells that could destroy it. Blocking PD-1 or PDL1 with antibodies unleashed the immune system against tumors in ways that were even more broadly effective than CTLA4 blockade. Drugs like nivolumab and pembrolizumab, you might have heard of Keytruda, that's pembrolizumab, showed remarkable results across multiple cancer types: melanoma, lung cancer, kidney cancer, bladder cancer, Hodgkin lymphoma, and more. In 2018, Allison and Honjo shared the Nobel Prize in Physiology or Medicine, and it felt like the culmination of over a century of effort that stretched all the way back to William Coley and his bacterial toxins. But checkpoint inhibitors are only part of the story. Because around the same time that Allison and Honjo were figuring out how to release the brakes on T cells, other researchers were working on something even more radical: the idea of engineering T cells themselves, redesigning them from the ground up to recognize and attack cancer. And the person most associated with that breakthrough is a soft-spoken immunologist at the University of Pennsylvania named Carl June, whose work on something called CAR T cell therapy began with a deeply personal tragedy. Carl June's wife, Cynthia, was diagnosed with ovarian cancer in the late 1990s, and she passed away in 2001. By that point, June had already been working on T-cell biology for years. He'd spent time at the Naval Medical Research Institute studying how to manipulate T-cells, partly in the context of bone marrow transplants and HIV research. But his wife's death sharpened something in him. It gave his work an urgency that went beyond academic curiosity. He has spoken about this openly, about how watching someone you love die from a disease that the immune system should theoretically be able to fight. That changes the way you think about your research. It makes you willing to take risks that a more cautious scientist might avoid. And CAR T therapy was, at the time, a very risky bet. The basic idea is this: you take T cells out of a patient's body, their own T cells, the ones that are already part of their immune system. Then you genetically modify those T cells in the lab, inserting a new gene that encodes something called a chimeric antigen receptor, or CAR. That's where the name comes from. A chimeric antigen receptor is essentially a synthetic molecule that combines pieces from different parts of the immune system into one engineered structure. The outside part of the receptor is designed to recognize a specific molecule on the surface of cancer cells. The inside part is wired to activate the T cell, to send all the go signals, the proliferation signals, the killing signals. So when you put these modified T cells back into the patient, they circulate through the body, and when they encounter a cancer cell displaying that target molecule, the CAR locks on, the T cell activates, and it destroys the cancer cell. And because T cells can multiply, a relatively small number of engineered cells can expand into an army inside the patient's body. Now, the concept of engineering receptors onto T cells wasn't entirely new. An Israeli scientist named Zelig Eshar had pioneered the basic idea back in the late 1980s, creating what he called T-bodies, T cells with artificial receptors. But those early versions didn't work very well in practice. The T cells would activate, sure, but they wouldn't persist. They'd do their job for a little while and then fizzle out. The key insight that June and his collaborators brought, along with researchers like Michel Sadelain at Memorial Sloan Kettering and Stephen Rosenberg at the National Cancer Institute, was adding what's called a co-stimulatory domain to the CAR. Remember how we talked about T cells needing two signals to fully activate? The antigen recognition signal and a second co-stimulatory signal? Well, the early CARs only provided that first signal. June's group and others figured out that by building the co-stimulatory signal directly into the receptor, essentially hardwiring both signals into one molecule, you could get T cells that not only activated, but persisted, multiplied, and formed memory. That was the engineering breakthrough that made the whole thing viable. The first really dramatic clinical results came in 2010 and 2011, and the story is remarkable. A man named Doug Olson, a retired scientist himself, had been living with chronic lymphocytic leukemia for years. His disease had stopped responding to chemotherapy. He enrolled in June's trial at the University of Pennsylvania. The CAR T cells they used were designed to target a molecule called CD19, which sits on the surface of B cells, both healthy B cells and the cancerous ones in leukemia. They infused the modified T cells into Olson's body, and then they waited. What happened next was both terrifying and extraordinary. About a week after the infusion, Olson became severely ill. High fevers, plummeting blood pressure, the kind of systemic inflammatory response that looks a lot like sepsis. His medical team wasn't entirely sure what was happening. It turned out that the CAR T cells were working almost too well. They were destroying cancer cells so rapidly and so massively that the dying cells were releasing enormous quantities of cytokines triggering what we now call cytokine release syndrome. And here's where all those threads from earlier come back together, right? The same cytokine storm phenomenon we talked about with the 1918 flu, the same inflammatory cascade that made COVID-19 so dangerous. It was showing up here, triggered not by a pathogen, but by the immune system's own engineered soldiers doing exactly what they were designed to do. The body's inflammatory response doesn't distinguish between good reasons and bad reasons for massive cell death. It just responds to the signals. June's team had to figure out, in real time, how to manage this. And here's where a bit of serendipity and cross-disciplinary thinking saved lives. One of June's colleagues, a pediatric oncologist, realized that the cytokine profile they were seeing in these patients, particularly the sky-high levels of a cytokine called interleukin-6, looked very similar to what happens in certain rheumatological conditions. And there was already a drug on the market that blocked interleukin-6. It was called tocilizumab, and it had been developed for rheumatoid arthritis. They tried it, and it worked. The inflammatory symptoms came down rapidly, and the CAR T cells kept killing cancer. That drug, tocilizumab, is now a standard part of CAR T therapy management. It's also, incidentally, one of the drugs that was later used to treat severe COVID-19 cytokine storms. The connections just keep circling back. But the results, the actual cancer results, were stunning. Doug Olson's leukemia went into complete remission. So did other patients in the trial. One of the most famous cases was a 6-year-old girl named Emily Whitehead, who in 2012 became the first child to receive CAR T therapy. She had acute lymphoblastic leukemia that had relapsed twice after chemotherapy. She was running out of options. The CAR T infusion made her desperately sick. She ended up in the ICU on a ventilator, and her medical team genuinely thought they might lose her. But they used tocilizumab. Her inflammation came down, and when they checked her bone marrow afterward, there was no cancer. None. Emily Whitehead is still alive today, over a decade later, with no detectable leukemia. She became the face of CAR T therapy, and her story helped drive the push toward FDA approval. In 2017, the FDA approved the first CAR T therapy, tisagenlecleucel, marketed as Kymriah, for pediatric and young adult patients with relapsed B-cell acute lymphoblastic leukemia. A second CAR T product, axicabtagene ciloleucel, was approved shortly after for certain types of lymphoma. These were genuinely historic moments. For the first time, the FDA was approving a therapy that involved genetically engineering a patient's own immune cells. It's not a drug in the traditional sense. It's a living treatment. Each dose is custom-made from the patient's own T cells, and that, of course, is part of what makes it both miraculous and enormously challenging. The manufacturing process is complex, time-consuming, and expensive. A single course of CAR T therapy can cost several $100,000, and it doesn't work for everyone. Solid tumors, the cancers that form lumps in organs like the lung, breast, colon, pancreas, have proven much harder to target with CAR T than blood cancers. Solid tumors create that hostile microenvironment we talked about, full of immunosuppressive signals, physical barriers, and a confusing mix of target molecules that make it hard for engineered T cells to penetrate and survive. So the field is still evolving. Researchers are working on next-generation CARs that can resist the immunosuppressive tumor microenvironment, CARs that target multiple antigens at once so cancer can't escape by simply shedding one surface molecule, and even off-the-shelf CAR T cells derived from healthy donors rather than the patient themselves, which could dramatically reduce cost and manufacturing time. There's work on using other immune cells too, natural killer cells for instance, engineered with similar receptors. And this brings us to something that I think is one of the most profound shifts in how we think about medicine in general. Because what immunotherapy has done, whether it's checkpoint inhibitors like ipilimumab and pembrolizumab, or engineered cell therapies like CAR-T, is fundamentally change the relationship between the doctor and the disease. For most of medical history, the paradigm was, find the disease, attack the disease directly, cut it out, poison it with chemotherapy, burn it with radiation. The treatment targets the disease. But immunotherapy flips that. The treatment targets the immune system. You're not attacking the cancer directly. You're empowering or engineering the body's own defense system to do the attacking. And that's a conceptual revolution that extends far beyond cancer. Think about where this is heading. Researchers are now exploring whether similar approaches could work for infectious diseases, engineering T cells to fight chronic viral infections like HIV, where the virus has evolved to evade natural immune responses. There's work on using modified immune cells to treat autoimmune diseases, essentially reprogramming the immune system to stop attacking the body's own tissues. There are even early-stage experiments with something called regulatory CAR-T cells. T cells engineered not to kill, but to suppress, to calm down overactive immune responses in specific tissues. Imagine a world where instead of taking immunosuppressive drugs after an organ transplant, drugs that dampen your entire immune system and leave you vulnerable to infections, you could receive engineered regulatory T cells that specifically protect the transplanted organ while leaving the rest of your immune system intact. That's not science fiction. People are working on this right now. And all of this connects to a broader revolution in how we understand the immune system's role in diseases we never thought of as immunological. Over the past couple of decades, researchers have discovered that chronic inflammation, that low-grade, persistent activation of the immune system, plays a role in an astonishing range of conditions. Heart disease, for example. Atherosclerosis, the buildup of plaques in your arteries, was long thought to be purely a plumbing problem, cholesterol deposits clogging pipes. But it turns out that inflammation is central to the process. Immune cells infiltrate arterial walls. They respond to cholesterol deposits as if they were foreign invaders, and the resulting inflammatory response actually drives plaque formation and makes plaques unstable, more likely to rupture and cause heart attacks. In 2017, a landmark clinical trial called CANTOS showed that an anti-inflammatory drug, canakinumab, which blocks the cytokine interleukin-1 beta, could reduce the risk of heart attacks in patients who'd already had one, independent of their cholesterol levels. That was a genuine paradigm shift. Heart disease as, at least in part, an immune disease. And it doesn't stop there. Alzheimer's disease, type 2 diabetes, depression. There's growing evidence that immune dysregulation and chronic inflammation contribute to all of these. The microglia in your brain, which are essentially the resident immune cells of the central nervous system, appear to play crucial roles in neurodegenerative diseases. When they become chronically activated, they can damage the very neurons they're supposed to protect. The gut microbiome, that vast community of bacteria living in your intestines, is in constant dialogue with your immune system, and disruptions to that dialogue appear to influence everything from inflammatory bowel disease to mental health. The immune system isn't just a defense force, it's a regulatory system that touches virtually every aspect of your physiology. Which brings us to something that feels almost philosophical, something about the nature of self and identity at the cellular level. Because one of the most remarkable things modern immunology has revealed is just how much of what your body does every single day involves the immune system making decisions about what belongs and what doesn't. What's you and what's not you? And that question, that fundamental, constant, molecular question, is far more complex than anyone imagined even a few decades ago. Think about it this way. Right now, as you're lying there, your immune system is surveying billions of cells. It's checking surfaces, reading molecular signatures, sampling the chemical environment in every tissue. And for each encounter, it's making a call, friend or foe, self or other, tolerate or attack. And the astonishing thing is that it gets this right almost every time. Trillions of interactions per day, and the vast majority of them result in the correct decision. You don't notice because the system works. You only notice when it doesn't. When it attacks your own thyroid, or fails to catch a tumor, or overreacts to pollen. But the baseline reality, the everyday miracle, is that this enormously complex system maintains a dynamic equilibrium that keeps you alive and functional without you ever having to think about it. And here's what makes the question of self and not self so much stranger than it first appears. You are not, in a strictly biological sense, entirely you. Your body is home to roughly as many bacterial cells as human cells. Your gut alone harbors something like 30 to 40 trillion microorganisms, bacteria, fungi, viruses, archaea, collectively weighing a few pounds. These organisms are not you, genetically speaking. They carry entirely foreign DNA. And yet, your immune system, for the most part, tolerates them. Not just tolerates them, actively cooperates with them. Certain gut bacteria help train your immune cells, calibrate inflammatory responses, even influence which T cell populations expand and which ones are kept in check. There are specific bacterial species that promote the development of regulatory T cells in the gut lining, the very cells that prevent excessive immune responses. So the immune system isn't just deciding between self and foreign. It's deciding between foreign and dangerous and foreign and beneficial. It's making nuanced, context-dependent judgments, constantly, at a scale that's almost impossible to fully comprehend. This is one of the reasons the microbiome has become such a hot area of research. Because when that dialogue between gut bacteria and immune cells goes wrong, when antibiotics wipe out key bacterial populations, or when diet shifts the composition of the microbiome, or when the intestinal barrier becomes more permeable than it should be, the downstream effects on the immune system can be profound. Inflammatory bowel diseases like Crohn's and ulcerative colitis involve a breakdown in the immune system's tolerance of gut bacteria. The immune cells in the intestinal wall start treating normal, beneficial bacteria as threats, and the resulting chronic inflammation damages the gut itself. And there's increasingly strong evidence that these disruptions ripple outward, affecting systems far from the gut. The gut-brain axis, as it's sometimes called, appears to be a real and significant pathway through which immune activity in the intestines can influence mood, cognition, even behavior. Some of the most intriguing recent work in psychiatry involves the observation that subsets of patients with major depression show elevated inflammatory markers, higher levels of C-reactive protein, higher levels of certain cytokines like interleukin-6 and tumor necrosis factor alpha. Not all depressed patients, but a meaningful subset. And in some clinical trials, anti-inflammatory treatments have shown modest antidepressant effects in these patients. It's early, it's complicated. But the very idea that depression might, in some cases, have an immune component, that's a conceptual bridge that would have seemed absurd 30 years ago. And this is really the larger story that modern immunology is telling us. The immune system is not a standalone department. It's not a security team operating independently from the rest of the organization. It's woven into everything. It shapes how wounds heal, how bones remodel, how fat tissue functions, how the brain develops. During pregnancy, the immune system performs what might be its most remarkable balancing act of all, tolerating the fetus, which is genetically half foreign, carrying the father's antigens, and yet somehow not triggering rejection. The placenta creates a kind of immunological détente, a local environment where specific immune responses are dialed down while others are maintained. Regulatory T cells accumulate at the maternal-fetal interface. Certain molecules on placental cells suppress local immune activation. It's not that the mother's immune system ignores the fetus. It actively recognizes it and then actively chooses tolerance. And when this process fails, when the immune system doesn't properly tolerate the fetus, the consequences can include preeclampsia, recurrent miscarriage, and other serious complications. So even the continuation of our species depends on the immune system getting its self versus not self calculations right, in the most intimate and high-stakes context imaginable. Now, if you've been following along through everything we've talked about tonight, from the ancient Greeks and their humors, through Jenner scraping cowpox into a boy's arm, through Mechnikov peering at starfish larvae under his microscope, through Tonegawa unraveling the genetic shuffling that gives us antibody diversity, through the heartbreak and triumph of Emily Whitehead's story, you might notice something about the arc of this whole journey. It's a story of increasing humility. Every generation of scientists thought they had the immune system mostly figured out, and every generation turned out to be wrong in important ways. The humoralists thought it was all about antibodies in the blood. The cellularists thought it was all about phagocytes eating invaders. Burnett thought clonal selection explained everything. Janeway realized innate immunity was far more sophisticated than anyone had given it credit for. And now, in the 21st century, we're realizing that the immune system is so deeply integrated with the rest of physiology that the very concept of an immune system, as a separate entity, might be a convenient fiction, useful for textbooks, but not quite capturing the reality of how the body actually works. And yet, for all that complexity, for all the things we still don't understand, the practical payoff of what we've learned is extraordinary. Think about where we started. For most of human history, infectious disease was the leading cause of death. Children died of smallpox, of measles, of diphtheria, in numbers that are hard for us to even process today. A mother in 18th century Europe might expect to lose half her children before they reached adulthood. That was normal. That was the baseline of human existence. And now, because of vaccines, because Jenner had an insight and Pasteur refined it, and generations of immunologists figured out how to train the adaptive immune system safely, diseases that once killed millions are either eradicated or controlled. Smallpox is gone. Polio is nearly gone. Measles, when vaccination rates are maintained, is held at bay. The COVID-19 pandemic, for all its devastation, saw the development of effective vaccines in under a year, an achievement that would have been literally unimaginable without the accumulated knowledge of how the immune system recognizes antigens, how T cells and B cells respond, how memory is formed. And beyond vaccines, the immunotherapy revolution we talked about, checkpoint inhibitors, CAR T cells, is saving lives that would have been lost just 15 years ago. Patients with metastatic melanoma, a disease that was essentially a death sentence in the 1990s, now sometimes achieve complete durable remissions with checkpoint inhibitor therapy. Children with relapsed leukemia, who had exhausted every other option are alive and cancer-free years after CAR T treatment. These aren't theoretical benefits. These are real people, living real lives, because scientists spent decades studying how T cells are activated, how tumors evade immune detection, how you can release the brakes on an immune response that the cancer has learned to suppress. But here's the thing that feels most important to sit with as we wind down tonight. The immune system, for all the engineering analogies we might use, and I've used plenty of them, talking about checkpoints and brakes and sentinels and surveillance, is not really a machine. It's something more fluid and alive than that. It's a system that learns, it remembers, it adapts, it makes mistakes and sometimes corrects them. It negotiates with the microorganisms that share your body. It shapes and is shaped by your experiences, the infections you've had, the foods you've eaten, the stress you've endured, even the people you've been close to. Your immune system is, in a very real sense, a record of your life. The antibodies in your blood right now are a molecular diary of every pathogen you've ever encountered and overcome. The memory T cells circulating through your tissues carry the imprint of battles fought and won years or decades ago. Doug Olson's modified T cells, still patrolling his body more than a decade after his treatment, are a living testament to a collaboration between human ingenuity and biological machinery that no one fully predicted would work as well as it did. And the thing that gets me, the thing I keep coming back to, is that we're still so early in understanding all of this. The field of immunology is maybe 150 years old, depending on where you start counting. The innate immune system's pattern recognition receptors were only identified in the late 1990s. CAR T therapy was approved in 2017. The role of the microbiome in immune regulation is still being mapped. We know enough to do remarkable things, but we're honest enough, the best scientists are anyway, to admit that the map is still mostly blank space. There are entire categories of immune cells whose functions we're only beginning to understand. There are interactions between the immune system and the nervous system, the endocrine system, the metabolic system, that we've barely scratched the surface of. So what does all of this mean for you, lying there tonight? Maybe just this. Your body is doing something extraordinary right now, something it's been doing every moment of every day since before you were born. It's maintaining you. It's keeping the boundary between you and everything else intact, while also knowing when to let that boundary flex, when to tolerate, when to cooperate. It's remembering every challenge it's ever faced and preparing quietly for challenges it hasn't seen yet. It's not perfect, no system this complex could be, but it is, by any reasonable measure, one of the most sophisticated networks that evolution has ever produced. Billions of years of trial and error, of organisms living and dying and passing on whatever worked, have built this system that hums along inside you right now, without your conscious input, without your awareness, doing its work in the dark. And maybe there's something comforting in that. In knowing that even as you drift off, even as your conscious mind lets go of the day and all its concerns, there's this whole other layer of intelligence, not the kind that thinks in words, but the kind that thinks in molecules and signals and cellular handshakes. That's awake, that's watching, that's keeping things steady. It doesn't need you to supervise. It's been doing this since long before humans had language to describe it, since long before anyone knew what a cell was or what an antibody looked like. It just works, quietly, persistently, in the background of everything you do. So let it do its work. Let your mind go quiet, the way your body already knows how to be quiet and busy at the same time. All those cells, all those signals, all that ancient, intricate, beautiful machinery. It's got this. It's been handling things since the very first organisms figured out how to tell the difference between themselves and the rest of the world, and it'll keep handling things tonight while you rest. That's what it's for. That's what it's always been for. And right now, that's enough. That's more than enough. Let yourself settle into that. Let the complexity fade into the background where it belongs, and just rest. You're in good hands, your own, as it turns out. Quietly, invisibly, remarkably good hands.